Go.
Greetings, everyone. I'm Mansi Agrawal.
And I'm Advait Krishna.
We are the co founders of Nurture Oncology, an initiative by which you will get to know recent updates in oncology research. Here, we are going to give you reviews and minutes from the recent articles in different aspects of oncology. And this is our very first review. So, we are picking up a paper from the aspect of molecular and clinical oncology. I hope you enjoy it. And let's begin.
Hello, everyone. Today we'll be discussing a paper, Multiple microRNAs as biomarkers for early breast cancer diagnosis by Jang et al. The paper is based on a novel approach towards early detection of breast cancer. As you all know, breast cancer is one of the commonest cancers worldwide among females. And mammography and USG are the early diagnostic methods. Mammography has problems such as false negatives, then it might warrant for unnecessary biopsies and radiation exposure is there, of course. And the sensitivity ranges from 62% to 89% and specificity is low too. Then for dense breast, the sensitivity is significantly lower and we go for ultrasound. But this also has problems such as a high dependence on test equipment and testers and then false positives cannot be avoided. Hence, it is necessary to develop a new and auxiliary test for diagnosing breast cancer in order to solve these limitations of mammography. The screening method. So they have touched upon several concepts over here. And this is one interesting thing, exosomes. Exosomes, most of us, we have read in biochemistry that they are groups of small membrane vesicles that are shed into body fluids or extracellular environment. And since exosomes are present in various body fluids and the stability of microRNAs are there in exosomes, exosomal microRNAs are another new class of biomarkers for early and minimally invasive cancer diagnosis. This is a really interesting point. We'll be sharing this article at the end, and then anyone, whoever is interested, please go to reference number 12 and read the paper upon it. Then, let's come to the methodology. In this study, 226 breast cancer patients and 146 healthy patients were used. There was the initial discovery study and a validation set. So in the initial part, 146 breast cancer patients and 90 healthy serum control were used. And then for validation, 80 breast cancer patients and then 56 healthy patients, healthy controls patients were plasma were used. And then it took place in Korea. Plasma samples from asymptomatic healthy donors were obtained. Exclusion criteria included known history of cancer, high-grade dysplasia, and autoimmune disease, to mention a few. After that, isolation of RNA was done from plasma. Total RNAs were extracted from plasma samples, and removal of genomic DNA and cDNA synthesis by reverse transcription was done. After that, Mann-Whitney U-test and Spearman's correlation test were used. To people who are beginning with research, statistical methods might look a bit foreign for you, but it's easy to learn. You can use any platforms such as Coursera, or there are workshops being conducted regularly by statistical teams from several institutes such as AIMS itself. And you can learn this online easily, whoever is interested in biostatistics. And if at all you need personal guidance, you can contact Mansi or me as well. And then before we get into the results, you must have an idea about what microRNAs are, how they're different from mRNAs. So Mansi will be elaborating upon the same right now. Over to Mansi.
Yeah, so Advait has been talking about microRNAs since the beginning of this article, but do we have a clue what microRNA really is? Now, to touch upon the role of microRNAs, they are entirely different from messenger RNAs. MicroRNAs are small regulatory molecules that bind to messenger RNA and prevent them from being translated into proteins. So this allows them to control the activity of various genes. Now, this study investigated pre-microRNAs, like the forms of precursor molecules that get processed into mature microRNAs. And they aim to identify these as potential biomarkers for breast cancer. Now, the researchers here focused upon 9 pre-microRNAs in blood plasma samples from breast cancer patients, as Advi told, and all these pre-microRNAs showed significant differences, which we'll look into the result parts. Now, this study directly, it's like interesting to note that this study didn't directly measure mRNA levels. Instead, they are indirectly pointing to the potential changes in mRNA regulation and gene expression associated with breast cancer development. So the microRNAs analyzed in this study, here's a list of the pre-microRNAs studied. There are 9 pre-microRNAs, and these are like microRNA 1246, microRNA 206, 24, then 373. Now, to give you brief of what the function of these microRNAs is. So, coming to the microRNA 1246, it is specifically limited research is there on microRNA 1246, specifically in breast cancer. However, some studies do say that they have a role in cancer development and progression in other organs like colon and kidneys. Now, microRNA 206 has been linked to tumor suppression in various cancers. Now, these microRNAs can either lead to progression of the cancer or tumor suppression. So some play a role in development and some in suppression, while some have a complex role which act as both. And one of these microRNAs is microRNA 24, which acts as a suppressor in some contexts, whereas it promotes cancer in others. So, and microRNA 373 is similar. It might function as a tumor suppressor in some cancers, but it can also regulate genes involved in cell cycle progression and apoptosis. So, coming to the significance, the researchers in this study measured the individual levels, then they went ahead with the combination approach. So the individual roles of these pre-microRNA are though not fully understood, and that's also potentially area of future research. But the entire significance lies in a combination that effectively differentiates breast cancer from healthy control. So coming to the how they measured the value of microRNAs, so they used real-time polymerase chain reaction for microRNA detection, and here they have denoted CT value in this chart, and on the x-axis we can see the both levels in cancer patients and the normal controls. Now, what is CT value? CT value refers to the number of cycles it takes for the fluorescent signal generated by the targeted microRNA to cross a predetermined threshold level. So this threshold level signifies a point where the signal is clearly detectable above background noise. Now, the people who requires lesser number of CT, lesser number of cycles to detect the fluorescent signal. So that means that they have more more level of microRNA, because less number of cycles are required to detect that signal. Whereas people or samples which have less microRNA requires more number of cycles, more amplification to generate that particular threshold. Therefore, here in all these charts, we can see that normal samples had a higher CT value as compared to the as compared to the cancer patients. Cancer patients generated the CT value earlier. Now, I hope you know the basics of TCR and you were able to understand why CT mattered. But you have to remember that CT provides a relative measure of the target microRNA concentration. It won't give you the absolute value. So, and this study found that all the microRNAs had different CT values between breast cancer and healthy control. So this figure you can find the attachment below in the description, and analyze these graphs by yourself. Now on to Advait.
So, thank you, Mansi, for the same. We'll go to the results part. Here, there have been previous studies conducted focusing on single microRNAs alone and their specific functions in specific situations as such. These are list of few papers based on the same. And then here, before going to the specific result, I have to touch upon the key points of what Mansi told regarding CT again. CT is just a cycle number and not an absolute measure of microRNA concentration. Lower the CT, it signifies a higher starting amount of microRNA. This is what you're supposed to keep in mind. And difference in CD values between groups can indicate changes in microRNA expression levels. So lower the CD signifies a higher starting amount of microRNA. Hope this is clear for everyone. Now, here you can see the training set AUC and the validation set AUC. The highest AUC was for these four microRNAs as such. 223, 1246, 206, and 24. And that means, let us come to the values. Yeah. So, these four microRNAs and their combinations, they were able to pinpoint that these four and their combinations achieved a remarkable accuracy rate of around 99.2% in differentiating breast cancer from healthy controls. This is a very promising finding, suggesting the possibility of a highly reliable blood test. That is a blood test for breast cancer detection. So, this result as such, based on these four microRNAs, showing that they and their combinations have a higher accuracy in determining breast cancer, was a significant finding. Along with that, dumbbell-like structural primer for pre-microRNA amplification by real-time PCR. This was a proprietary technology by their team, and that could minimize non-specific PCR in real time. This was used in the study. These two are the significant things which you can get from the study. And then, in conclusion, set of biomarkers developed in the study showed high accuracy for early diagnosis of breast cancer, and then it can be used to detect breast cancer at an early age. Plus, yeah, they conclude saying that these diagnostic indicators implemented can be implemented, hopefully, to address the problems of existing assistive methods for conventional mammography and effectively detect breast cancer at an early age. Hence, we've concluded the study. Thank you all for patient listening. Thank you very much.
Thank you, everyone.